OK, bonjour tout le monde. On va commencer tranquillement. On attend des personnes à l'œil. Donc, c'est un grand plaisir qu'on reçoit aujourd'hui Dr. Tokuriki de l'Université de Colombie-Britannique. Nobu est professeur de biochimie de son laboratoire et à l'Institut Michael Smith sur le campus à UBC. J'ai eu la chance de visiter son laboratoire l'année passée quand j'étais en SAVC. C'est super bel endroit pour faire de la science. Nobu a fait une formation qui était un peu loin de l'évolution au départ. Il a fait un doctorat à l'Université de Sakura dans un département qui était plus en ingénierie, donc il a travaillé en biophysique. Et ensuite, il est allé faire un premier stage postdoctoral en Israël, à l'Institut Weizmann, où il a travaillé avec Dan Tokuriki sur l'évolution des enzymes. Et c'est là qu'il a commencé vraiment à travailler sur l'évolution moléculaire. Puis ensuite, il a fait un deuxième postdoc à l'Université de Cambridge, en Angleterre. Il a terminé au début des années 2010, puis il a commencé ensuite sa carrière à UBC comme chercheur. Maintenant, il est professeur titulaire à UBC. Donc aujourd'hui, il va nous parler de ses travaux de recherche. Nobu fait des travaux en évolution moléculaire en utilisant les enzymes comme modèle, puis s'intéresse à la relation entre la séquence, la structure des protéines, et leur capacité à évoluer vers de nouvelles fonctions. Donc, il fait déjà dans ses travaux avec Dan Tokuriki, à l'époque, il avait déjà abordé ces sujets-là. Puis, un de ses papiers de postdoc, par exemple, qu'il a publié en 2009, a été cité 800 fois. Donc, c'est un des chercheurs, je dirais, en évolution moléculaire expérimentale, pas le côté de bioinformatique, le côté expérimental de l'évolution moléculaire, comment les mutations affectent la fonction des protéines, comment elles changent la fonction des protéines, puis comment ensemble elles définissent des nouvelles fonctions. Je dirais que c'est un des chercheurs les plus influents de sa carrière, définitivement. Donc, j'étais content de savoir qu'il pouvait venir en personne nous donner un séminaire. Aujourd'hui, il va nous parler de l'evolvabilité des protéines. Thanks a lot again for coming.
Thank you very much. Thank everyone spending time with me in the morning. It was nice to hear your works, and I'm looking forward to meet some other people, maybe Sophie, and looking forward to your defense. I thought I just prepared a nasty question. Talk. All right, so then I put a nice picture of the mountains. There's the Whistler around the Vancouver. If you're interested in outdoor and mountains and rivers, you should check the UBC. This is a part of other side of the Canada, but there's lots of nice research. I'm always looking for good people. So if you're interested in our research, and just let me know. So since my PhD and postdoc and then being the PI in the UBC, I'm interested in how protein enzyme evolve. I'm generally interested in how the evolution happens, but it somehow started from the protein lab and doing the biophysics. I'm stuck in the protein enzyme. And so that's the big concept of simply how new enzyme evolve. But if there's the very vague questions, and then some people don't think this is a science, so then I just need to go down to a little bit more specific of the questions. That's why some of the list that I'm asking in terms of research. So that way we getting into a little bit not only the protein levels, we try to get in the metabolic pathways and more like a microbial community as well. So it's and try to expand what's the evolution happens. But today I'm more or less focused on the topic that's the protein center. And then the topic is epistasis, like a mutational interactions, and then how this mutational interaction affect the protein evolution. And then I will show you some of the example that's maybe not perfectly, but we try to understand why this is happening. So this is my sort of typical picture you've seen in sometime in evolutionary talk. Well, evolution can depict in a very simplistic way. It's maybe not reflected in the real world, but you can draw the sequence space of the proteins, organisms, whatever, and evolution, you can think about climbing the mountain, right? So then the question is how this mountain or the geography is look like. It's like how it's locked and smooth. If it's smooth, and there's no simple there's no complications. And if you have good mutation, you can collect 20 beneficial mutation, combine it, you get the best solution. That's a simple solution in evolutions. But then if you have bit of epistasis, lagness, you maybe end up like local maximum, lower fitness peak, and that's what you want to avoid it in engineering side. Evolution somehow escaping the making some of the good enzymes. So the simple question is how easy to reach the adaptive peak, right? So if you do the engineering, how easy to engineer the enzyme? That's the same questions, and how we kind of messed up our system. If you do try to engineer, then why we not able to succeed? That's kind of my question started from the protein engineering field. That's people frustrated myself with there are lots of failures. Even we don't publish, that's failure. So the concept is I'm always talking is epistasis. So there's a very simple schematic here. There's if you have the two mutation, one mutation input like threefold, another mutation input twofold, and if you combine those in additive order, you should see the sixfold improvement. Right? It's like a log scale additive effect. So or you can multiply them. So that's kind of the no interaction between mutations. So that's the simplest way. But if you have interactions, my two mutation can be like eightfold effect instead of 6, and that's kind of synergistic positive epistasis. And if you have sort of antagonistic interaction, then the effect become lower, fourfold, threefold. And in some cases, start to see the negative effect. So that's the more complicated situation. If you put the two positive mutations, when you combine it, and then become a negative. That possibly happens, and then that's obviously you can see that's evolution. Complicated, but other cases, the one mutation is negative, one is positive. When you combine it, can be both effect is synergistically improved. So that's obviously complicated evolution. So that's kind of the cause of the mountain shape and the lagness happens. Then what we want to do is try to get into the molecular level and try to understand it, and hopefully we can predict this type of interaction causing the disease. So I roughly follow 3 topics today. How much epistasis exists? We always say a lot of epistasis, no epistasis from the paper. I want to quantify it. So I will show them quantifications, and then I will show you how this, by certain case that how epistasis affect the protein evolutions, and I will show you some of the case that we little bit understand how the molecule works. Okay. So the first one, I just wanted to talk about how much epistasis exists. So there are lots of literatures, and it's confused everyone. And then because of the different system studies and a different target, and obviously every system is different, and what people looking at the different as well. So when you look at very large scale study, let's say take the protein, mutate every position, single point mutations, and the people you can do it every combination of the mutation, every protein. So it's kind of protein scale epistasis analysis. And that's actually, you don't see many epistasis. Because that's why most of the cases, epistasis happen with very interactive residues. Now you know the alpha-4-2 can predict your protein structure. That's actually coming from a coevolution analysis. But a coevolution analysis is essentially telling you which residues are interacting. But a very strong signal of epistasis usually coming from the contacted residue. So if you do very large scale, actually, you may not see much of epistasis. But when you look at actually what we usually looking at the enzyme for protein evolution side, which actually mutation has to change the function. That's what we are looking at. So then the mutations usually accumulate around the active site, and the mutation changing the way that enzyme and the substrate interact, or the catalysis happens. So in this case, we see a lot of epistasis. When you see the literature, there's people generalize quite a bit, but this what we are seeing. I just wanted to clarify, we are looking at the really adaptive process and the mutation collectively changing the function. Okay. In that case, there are a lot of epistasis being observed because of sort of non-linear relationship between different property of the proteins. So this case is a very simple schematic. So this, let's say, resistance level of the certain drug, like EC50, effective concentration. Protein, and then let's say the growth rate of the bacteria or the microbes. In that case, it's like if you have very strong proteins, it doesn't change the fitness because it's good enough. And the very bad, and you start to see the obviously, right? Like on the bottom line, you don't see any flow. So it's like this usually this behave like a sigmoidal. Protein folding is behaved sigmoidal. Because maintain actually, if you plot in the log scale, it's sigmoidal. So every biological system we can draw in a sigmoidal shape. And then if you look at this phase, obviously it's a kind of linear phase. Some experiment you're doing is like a linear relations. You can see it, but if you expand it million-fold change in substrate concentration, ligand concentration, you see in the sigmoid. Okay, this case, so you always, whatever you do is you see the epistasis, one mutation increase a bit, other mutation decrease a lot. But if you combine it, actually, it's still decrease, but it's not as additive as you expect. So this type of epistasis, obviously, you see it. It's not change the sign, so that's important. The message. It changed the degree of epistasis, but it won't change the sign, right? Because it's the direction. So that's happens, but I don't want to talk these things because it's also complicated and different system. We want to extract sort of specific epistasis. That's this parameter changes. Okay, so that's only the parameters we want to. Roughly, that's what we want to do. Okay, so that's adaptive mutation. That's what we want to look at it. So the first kind of pioneer work of this epistasis is coming from the Dan Beinlich and Dan Hartle and from Harvard. So they have the nice experiments. This TEM1 beta-lactamase enzyme is if you use a plasmid of beta-lactamase or the ampicillin resistance, this enzyme is located. But this enzyme is actually very important clinical enzymes, given the resistance to the bacteria. Also, this enzyme evolving over the time since we started to use the penicillin in the clinics. And then some point, people found the mutants has 5 mutations, and that's increased resistance level about 4,000-fold against the type of the antibiotics called cefotaxime. So the third generation cephalosporin is slightly different from the ampicillin or penicillin. And then they knew this is the kind of resistance mutant. So the Dan Beinlich took these mutants, and they made all mutants. So if you have 5 mutations, you can make all the 5 combinatorial mutations, single, double, triple, quadruple, and all the mutation accumulate. And then test all this resistance level, and then measure it, and then see now you can estimate what's the fit. So this diagram actually that doesn't show everything. If the activity goes down, they didn't show the diagram. So that's out of the 5 mutations, only 2 mutations improved activity from the wild type, another mutation. Message is like a single white point state. You can go up to with 5 mutations, but first point only 2 mutations is useful. So the 3 other mutations somehow become positive after the 2 mutations. Okay, so they calculate sort of pathway analysis from the starting point to the last point. There's 120 pathway if you calculate everything, and only 18 pathways possible to reach throughout the uphill evolution. So that's why you can see they didn't do much of analysis. So we want to get into more statistic. So this is actually the way if you do the experiment, you can see this. Right? The 5 mutations, single, double, triple. You can measure it, analysis. We are not the first one doing it. The people started to do the most systematic analysis and statistical analysis to see what's the effect in this fit. Okay, so that's what we wanted to do. So that we want to look for some new system. This is the one example is called the methylpalathion hydrolase or MPH. So this enzyme is very interesting enzyme because it's hydrolyzed organophosphate. So organophosphate is human synthesized compound that people use for the pesticide. So the German scientist discovered this way to synthesize it. And then it's happened to be a pesticide, and the people excited to use a lot. If you modify this compound a little bit, then you can make nerve gas as well. So this compound binds to acetylcholinesterase. This one is particularly to the insect, but it's obviously a little bit bind to the human ones too. So now it's banned, but it's used a lot in everywhere in the world. Because we use it a lot, and the bacteria start to evolve the enzyme to degrade it. So the people realized actually when we started to spray it, oh, it's nice pesticide. After 10, 20 years, it's not working as much as they wanted. And they started to study what's happening there, and actually soil bacteria has enzyme degraded. And they breaking down this compound, take the phosphate out, the carbon out, and they are using that as a nutrient. Okay, so that's the evolution that's supposed to happen very recently. That's kind of a nice example for us for the enzyme evolution. So this is when you draw the phylogeny, this is the enzyme I'm talking. So there's the 4 different substrate here, and you can see that very active. When you look at the homologous enzyme, there's a cyan color, is kind of dihydrochumulin. So there's a different reaction. Lactonase reaction that seems to be the most preferred substrate for the dishomologous enzyme, but this enzyme is obviously still the promiscuous activity has shifted. Okay. So still that sequence difference about 100 amino acids. So we want to narrow down what's mutations causing this evolution. So we did ancestor reconstruction. Essentially, software predict sequence of most probable sequence of this node, and takes this sequence, synthesize it, characterize it, and you see why cyan bar is very high. It seems to be to the other ones, promiscuous activity, whether this organophosphate is relatively high, but we like it, right? Because it's ancestral. Maybe it's just predicted, not real ancestors, but it has a promiscuous activities, and it looks like ancestral state. Okay, and then there are still the 32 mutations, so we want to narrow down further. So we simply look at the structure and also the sequence alignment as well, and identified 5 mutation in the active site. We started from 4 and then added 1 mutation, and then see how these mutations change the activity. We tested. So you can see we're testing the 5 mutation ancestral state to the making to the 5 mutation to the MPH, MPH back into the ancestor. Okay, so it's simply testing the 5 mutations. It's easily, you can see that it works really beautifully. The ancestral state with 1 mutations, it becomes like an MPH function. MPH 5 mutation reverse it just becomes ancestral state. So we're very happy about it. I'm not sure this is really happening in nature. It doesn't matter for me. You can see this 5 mutation and change the function of that. Okay. So, okay. So we solve the crystal structure, or our collaborator solve the structure. It really makes sense. This ancestral enzyme, this like MPH, you can see the pocket become larger. Now it's fit to this parathion. If you just place this parathion the same way in water, it's obvious the hindrance happens and it doesn't work. So it looks like really explain active site change as well. Okay, so now 5 mutation, it's nice, it's the same as the Dunn-Bailey's experiment. So we can do this experiment, make the order combination of the mutations, and about 1,000-fold improvement in activity, and see what's the activity changes. So this is what we get. So this is the thick line. It's kind of most probable uphill evolutions. First mutation improved quite a bit, and then second, third doesn't, and then fourth, fifth. But it's you get 1,000-fold improvement sort of uphill way. So it's explained the evolution could happen as well. Interesting one, this 271, it's like 5-fold negative when you test in the water type, but somehow it flips somewhere in between. So actually there's the 2 or 3 mutations needs to fix it to become these mutations become positive. But it's obviously changing the effect to be positive, and then 5 mutation is the best one you can see. So we can do a little bit of statistics. So what we do is actually, when you look at this map and then look at the single point mutation, is actually a single point mutation can happen 16 times in this landscape. From the wild type, from other, on top of other mutations. You can extract those mutation effects and then see how this distribution happens. Right? So this a kind of try to look at how much epistasis effect happening. So this is a kind of the result. So it's very interesting here. For example, this is a wild effect, and some wild type is obviously 10-fold improvement. Some gets like 100-fold, and some cases become the negative. And these two mutations are more or less like a binary pattern. So the kind of some mutations responsible for certain behaviors, mostly positive, but you can see those other mutations change the sign, right, from the positive to negative. You can also analyze double mutation effect. So the double mutation happened 8 times in this landscape. So if you look at the two mutations, 72 and 193, and then you have the double mutations, and you can simply calculate how much deviation happened from predicted additive effect to the real epistasis. Okay. So this kind of the statistical method that Dave Anderson, former postdoc in my lab, came up. So this is a little bit complicated graph. This bar represents single point mutation effect on average. So you have to average every mutations. And the width actually telling you how much deviation happens in single point mutation effect. So this is relatively smaller ones. And this is a second order mutation effect. There's some epistasis factor. On average, you can see positive and negative. This is third order, fourth order. And this explains how much fit you can get it using this average effect. Which means if you take this average effect of the first order, try to fit entire numbers within the fitness landscape, how much explanation you can get, okay? So simple message here is like if you consider up to second order, you can explain 90% of the fitness landscape. If you take in average, that's the point. So it's like, if you analyze actually this type of things, and usually you can get more satisfaction with second order. If you include the third order, it's almost get to the perfection. So you don't see that's why too many is going on from the average side. We can plot those effects in crystal structure or 3D structure. So this all represents sort of single order effect line in between second order and then third order effect. So you can see how the mutations are roughly interact. The substrate is supposed to be sitting here. Right? So this mutation is obviously not going to interact with other residues physically, but probably through the substrate, it does interact. So you can do same experiments with 4 different substituents. You can see top one, bottom one difference is the oxygen here or the thiol group here. There's the side chain, the methyl group and the ethyl group. There's a tiny difference in substrate actually changed a bit of the fitness landscape. Very difficult to see, so we can map in the structure. So you can see here, if it's the thiol here, this is actually the average positive, oxygen is negative, and the interaction between those also changed quite a bit. We don't know exactly how it's working, but obviously this substrate sitting here, so the oxygen possibly around here. So that's the interaction probably happening to the between substrate and the next one. So we, unfortunately, we didn't understand more than this. So we done crystal structure MDs and stuff, and it's very messy. Matter flying out, substrate flying out in MD. So we can't really tell what's exactly happening, but it's like a other way to see, well, if you do this type of mutation analysis, actually you can see how enzymes interact with the substrate. Okay, so this story I just wanted to show. Okay, so there are lots of epistasis on this, right? pH. Obviously, you're gonna ask also what? Okay, so we want to see a little bit more statistical picture. Then we want to see why we can do this, right? Use these ideas of the engineering. So grad student in my lab in Carl, I started to collect all this literature. So we restricted the data set that's enzyme evolution, people done adaptive evolution. So many of them coming from my lab, some other group done it. but it's not looking at the protein interactions, or protein folding, or like a mutation that's randomly selected. But we just focus on the directed evolution or evolutionary examples. But people done in the different substrate, different mutational set as well. So it's actually expanded quite a bit of data sets, like 7 enzymes, but 56 positions in total. And then people, because do it in different substrate and different environment, we done different metal substituent as well. So it's you get quite a bit of data set. So first thing that we just analyze simply is this sort of deviation or heterogeneity of the single point mutation effect. So simply calculate 2 times SD, and then how the distribution happens in single point mutation effect. So there's a kind of different data set. Obviously, each system is different, but we just wanted to see the distribution. So it's amazing things you can see. This is actually 10-fold already here. So the many of the system actually 100-fold, 1,000-fold deviation when you look at the single point mutation in different genetic backgrounds. So somehow epistasis is happening. Only a tiny fraction of the data set is less than 2-fold. That's probably you think it's an experimental noise. But the many systems actually epistasis. We calculated obviously when negative or positive, only tiny fraction of mutations stayed in exactly same sign or neutral. The other one is change the neutral to negative or positive. More than 50% actually change the sign of the effect. So obviously epistasis is changing quite a bit of what mutation behaves. We also analyze how the wild type effect, because kind of it's important for the engineering or evolution, when you always start from certain sequence. When you look at the data set from wild type, how much explains average effect. When I told you the average effect nicely explains more or less the landscape. But when you look at the wild type, it's deviate from the average, obviously, it's just going to start to fade it. So how much deviation happened? You can see that, right? Not as much as distributions, but the average deviation from the wild type to the average is kind of there. So it's epistasis there. Even you look at the right reference point, there are some deviations you see. It's always from the entire picture. So this is the data set for the second order epistasis, so how the mutational effect distributed. So you can see the pretty much same picture. There's so much deviation as well as sign of the epistasis change all the time. And the wild type effect really doesn't perfectly match with the average. If you do the third or the fourth, well, actually you can see the same things. So, okay, so it looks like mass. And then we were just wanting to see how messy it is a little bit more. So what we do is try to predict mutational effect and last variance using a limiting data set. So I show you, if you use average data set, second order, third order, we will explain everything. But what we want to calculate is simply very more primitive way. Let's say if you have the single point mutational data, how much you can predict these numbers, believing this is additive effect. Right? Then you can do, if you have a double mutational data, now you can include the epistasis factors, but it's coming from the wild type background and how much you can explain. The bar graph looks messy. So some of them looks okay. Right? So if you have black one is actual data set, and then the red one is include every data set. So obviously your prediction is perfect, but this maybe this one is chaotic. So if you have single point mutations, data is very bad. Double mutation is actually nicely fit. But actually, if you include in the more factors, it actually start to deviate it because it's just so random things going on. So the predictions become so weird, even you include the second order terms. Right? Sometimes it works, sometimes it doesn't. For example, this data set here, single point mutations kind of underestimate. If you include double mutations, overestimate, and then third order is start to settle. So this is obviously telling us very difficult to predict because of the certain interaction happens. Obviously, set of the mutation is there. Right? if you look at only the data set, it's very, very difficult to predict what's happening. So there's kind of the messy situations, but I just wanted to see it. And it's obviously that's something we have to consider, obviously predicting something or designing something. So it's probably the key is underneath like a molecular mechanism, actually how the residue interact. It's obviously not easy. Obviously, if you have a data set, it's better. It's better than nothing. So you can do some predictions, but you may not get the perfect prediction to do. All right. So the second half of the talk, I want to talk about a little bit more examples. Why this happens, and then evolutionary outcomes. The one experiment I did starting from my PhD is evolving the one enzyme. So I always wanted to change the enzyme one to another and see what's happened. So this example is a phosphotriesterase, the PT. It's a similar enzyme to the MPH. It's degrade this paraoxone organophosphate. So it's the same type of enzyme, but the evolved independently. Again, it's found in some bacteria, but the hydrolyzed paraoxone. The parathion loves this S and at the point. This one is O is the preferred for some reason. So I took this enzyme and then randomly decide this reaction to target because this promiscuously hydrolyzed. This is the esterase, ester substrate, so it's the esterase reaction. So what's the difference in here is a PO bond cleavage, and this is a CO bond cleavage. Okay, so it's a chemically different center. That's why I kind of like to see the transition, but the hydrolytic reactions and water attacks and break the bond. So I did the directed evolution 18 rounds. Take the enzyme, mutate it, screen it, take the best enzyme. Sometimes we do the shuffling. Anyway, accumulate the mutation over the time and try to improve as much as possible. Okay, so there's 18 rounds. You see starting the plateau, getting bored of it and then tired of it. So I stopped in 18. Actually, we went to the 22, but it's almost, this is plateau I can reach. The enzymes become, KK over KM is very, very close to diffusion limit. So this is a nice example of the enzyme evolution, 10,000-fold activity improvement. And the mutation accumulated in the active site. The red colors are the kind of fast round mutations, and the green ones that are outside, and the later round of evolution. So it kind of makes sense. Fast mutation improve a lot, and other mutations gradually accumulate. The important mutations in this talk is this mutations, 254R. The original amino acid histidine changed to arginine. We know this mutation is very important. And it's key activity. Sorry. So this that's the structure of the enzyme. It's the water-type enzyme with paraoxone. Nicely, you can see the pockets in here. And this is the evolved enzyme now. This is the plain hexanide substrate sitting in a nice, like a sort of narrow pockets. And this arginine 254 is actually sitting next to the naphthol group. Okay. So then we wanted to see some interesting evolution when we started in UBC. So we look at like a 254 positions, wanted to see some alternative evolution pathways. And look at the substitutions. We found 254 serine is kind of sort of neutral compared to the wild type, different amino acid. So we want to fix this serine instead of arginine and do the directed evolution. Okay. There's a bit of trick there. So that this setting we use needs to have 2 nucleotide mutations to change the arginine. So that when during the evolution doesn't change the arginine to go. So we want to do the alternative evolution pathway. This is what Charlotte did it. And you can see evolution kind of stop in the middle. Okay. So she pushed it quite a bit, but it doesn't change it. There's 100-fold activity difference starting from a different single point mutation. So the mutation is accumulating sort of interesting. Many mutations are different. You can see this is kind of a diagram. But 4 positions, we have the same positions being mutated. So 254, we intentionally change. Arginine is actually coming in the directed evolution, but serine is we chose 3 or 6 sitting there. It's exactly the same amino acids, isoleucine. But 233, 272 are the different mutations there. But then I can see this actually they clusters together. Okay. So that structure is very interesting here. So this is the mutation evolution example I showed. Arginine is sitting here, substrate here. Now the new evolution we did, substrate flipped to 54. Now you can see is there quite different geometry here. I just wanted to make it bigger. So you can see this evolution seems to be happening like making a very tight hydrogen bond network and actually very stable, stabilize this loop 7. And this evolution case is nothing interact because setting is very short. But this used to be aspartate, now arginine. There's no way to interact, and the loop just gone away. So you can't see in crystal structure, and seems to be very, very flexible. Okay, and then consequently, substrate is sitting a different. So this kind of you see the very best optimized evolution solutions, and a kind of intermediate, like it's obviously better than the water type. It's kind of local maximum solution here. So loop, I said like a loop 7 become very dynamic in serine trajectory we call, and arginine trajectory is actually very stabilized over the time. The dynamics changed, active site architecture changed, and obviously substrate binding changed. So we want to believe this is kind of 2 different peaks, or we want to call it even a molecular species. So we tried to test like a different mutational effect and see how it looked like. Okay, the first test we did it is mutating this key position back to the water type amino acid and see what happens. You can see every mutation we tested, the combination of those, everything is deleterious, which it makes sense because those mutations are accumulating during evolutions, mutating back, which is become deleterious. But another interesting experiment we're swapping the mutations. Okay, so there's the we get these mutants and then mutate every single point mutation to serine 1 and serine 1 mutating back to large. So you see that when mutating R18 to every position serine, it becomes very deleterious. And the interesting thing here is even changing in serine to the arginine positions doesn't improve it. Right? It's even a deleterious. So it's kind of look like it's really they become a different independent. Okay, so we want to get a little bit more. So the question is, well, it's okay separated. Really, when it's happened, these separations. Right? So single point mutations kind of dictate everything, or multiple mutation effect is there. So the structure, we solved a bunch of different structures and see what happens, and this is kind of ensemble refinement. So it's kind of analyzing the dynamics from the crystal structure. And you can see R18, you can see this loop 7, and the color is getting in tighter and tighter. So the gradually actually this dynamics is going down. And this one is gradually this loop quite more dynamic. And after this, we don't see it. So there's some kind of sense that this evolution is happening in a gradual way, because this single mutation doesn't do it in structures in a drastic way. So again, you can see this going down. This is actually MD data, and then we look at this is B factor data, and then just going down and then gradually going up in another. This 254 is important residues in interacting with substrate, and arginine actually is going down gradually as well, sending a little bit of the noise, but it doesn't do it. We feel like there's some kind of graduality going on. Obviously, we can test it in mutational way. So because of the loop 7 is dynamic so changing, we simply delete loop 7 genetically about, I don't remember what's the number of the amino acids, like 10, probably 10 amino acids being removed. So when you started from living from water type, it doesn't care the loop exists or not. And you can see as evolution goes, they start to feel the pain more. You can see those mutational effects happening in a very gradual manner. A serine initially is actually deleterious, but then it started to not to care at all. So you can remove the loop from the last variant of the evolutions. It is as active as the evolved one. So we can do the same thing to 54 positions, change it to the setting. So it's the kind of changing different directions. So you can see not single event happens and a gradual happen. Right? I talk about 4 mutations, but actually about probably 18 mutations happens here along the evolution, and that's what 10 mutations happen in setting. So it's everything is sort of gradually changing because it's a sort of network effect. So that's kind of interesting example of the sort of evolution can bifurcate. So this is what starting from a single mutation effect, and then obviously direction probably change, the preference of the Y mutation change. But if you accumulate in the more and the more, actually every mutation is contributing to shape this active site. And it seems to be that's the case. That's really the separation. You see it and after. Yeah, so that's rewiring definitely happening here. So this what simple things we can see is kind of forming the network, this kind of destroying the network. But that's not happening only those mutations, most likely. Other mutation in the loop 7 and other positions, it seems to be contributing this network. Yeah, so we just wrote like this. We see those kind of stuff, and we just wrote the review a couple of years ago, and I just wanted to show. So obviously, this is a very complicated system. Right? The protein has several hundred amino acids, and every atoms are moving together and then interacting together. And you obviously see certain interactions. And then it's not like when you look at some literatures, people analyze certain interactions and crystal structures and see how much effect happening in epistasis. So there's an example. This is our case, for example. Simple explanation we can have is 254, 233, it's interacting positively. So the 254, when it comes in, only 5-fold improvement. If you test actually 233 D2E on the back of the wild types, only 2-fold improvement. But if you combine actually 100-fold or 1,000-fold improvement in a synergistic way, because actually mutation 233 to the E is actually stabilizing the position of the heart. That simply happens. So there's kind of direct interaction between the substrate, between the residues and to the substrate. But other cases, there are many. So this example, mutation 2 actually coordinate position of the cofactor. So the obviously cofactor interact with the substrate. That's a piece that's been reported. This other case, actually, this is, I think, the DHFR. The 3 mutations here and there actually really doesn't interact each other, but it's look like they are accumulating around substrate. And also, it's not the, but it's kind of terpene synthase. Then probably each mutation interact with the substrate, and then probably changing the position of the substrate active site. So if you look at the crystal structure without substrate, probably you don't see anything, but it seems to be that's happening. Other cases, the dynamics obviously very important. This other beta-lactamase enzyme case, because of the famous loop in accommodating substrate, dynamics is very important for. And then those mutations in dynamics actually sometimes are far from the more drastic cases in the hemoglobin here, sort of allostry going on with one oxygen bind to the one unit, changing the white affinity to the other unit. Or those mutations actually scattered across the protein structures, but somehow allostry going on to do the epistasis. So obviously, there are different interactions happen in the proteins. And unfortunately, it's a kind of case by case, and unless you understand the enzyme or protein system, it will be very difficult to understand. Just last talk, I want to just show the other mechanism, probably not probably the structural. It's probably happening in epistasis. So this collaboration between Christopher Flori, he came as a visiting student from the University of Tromsø, Norway. So he came to my lab and doing a bit of experiments before the COVID. Then he came back and finished the work. So he did a directed evolution of one of the beta lactam called OXA48. So this enzyme degrades beta lactam antibiotics, but this one particularly doesn't like ceftazidime. This other third generation of cephalosporin, but it hydrolyzes ampicillin, penicillin, cephalosporin, other cephalosporin, and the carbapenem. So it's a kind of broad specificity enzyme, but somehow it doesn't like it. So he did a directed evolution, very similar way you can plate on resistance, agar plate and the resistance. Antibiotics contained, so the selection is very easy. He done several rounds, and you can see resistance improved about 100-fold. And he sequenced, and 5 mutations accumulated. And there's a 5 mutation accumulated. So this is the active site serine here. Substrate is supposed to be sitting here. And then this mutation comes fast, and other mutation comes later. You can see this is closest to the active site, and another 5 mutations. We think this one is not doing anything. So we take the 4 mutations here, then Chris did my favorite, this diagram. So there's an interesting situation again. This is the first mutation, 72, is only the one improve the resistance. And after that, but those 2 mutations doesn't do anything on top of the wild type, but those 2. And when you combine, it's actually the 5-fold improvement from here. So there's obviously strong synergistic epistase happening. And the combination goes a little bit farther resistance improvement. So what this situation is, again, this fast mutation seems to be a gatekeeper. I mean, it's kind of opened up the other mutations and accumulate. Without this, nothing happens. So this is kind of strange enzyme. Probably people in here is not enzymologists, but you can see probably how strange this enzyme is. So this is a time, just measuring the time of the reaction. This is the product per enzyme. This kind of it's supposed to be straight in the most of the case. If you incubate, reaction happens. If the enzyme is multiple turnover enzyme, then it just goes straight up. So you can see a little bit of kind of going down here, right? Maybe it's kind of experimental noise and whatever, you ignore it. But once you do the evolution, it's kind of this kind of kink happens so strongly. You can see this number is just like 8 and 4. So that means this enzyme's about after 4 turnovers, it's really getting slow. It doesn't get inhibited completely. It's just continuously going the reaction. If you kind of stop the reaction, put in the flesh substrate, it actually burst happens again. There seems to be some kind of weird burst happen in this enzyme. I don't know exactly how it happens, but this kind of strange behavior happens. So the kind of 2 biphasic behavior of the enzyme kinetics. So we can do obviously enzymatic Michaelis-Menten kinetics on burst phase and then steady state on the second phase. Okay, this is the burst phase kinetics. You can see nice lines on the roughly fit to the equation. Some reaction is very straight because it's the Km is very high. But you can see this, the burst phase kinetics really explains improvement of the resistance level in bacteria. So maybe this makes sense because the fast phase, you can degrade amount of the antibiotics, then that's enough for the bacteria because we don't keep supplying the antibiotics in the selections. So the fast reaction is enough to give you the resistance, and slow phase, they don't need to do it. So that anyway, so the burst phase seems to be do the reaction. Then there's the kinetic parameters. So you can see this fast mutation of the Km quite a bit. So Km is a sort of binding factor to the substrate. Okay, so the one doesn't bind to the substrate, then some number we can't fit the entire equation. But the fast mutation 72 lower the Km is increase the binding. So somehow access to the substrate under the catalysis. And those two mutations, by itself, it doesn't do much, right? A little bit, probably twofold improvement at most, maybe it's just noise. But when it's mutation on top of this 72, you can see even KM drops further, maybe it's a bit noisy, but this KCAT clearly improves. So it's kind of catalysis happens, but it kind of makes sense, right? When substrate has to come in to the catalysis, if you just mutate the white residues, just potentially improve the catalysis, but if the substrate is not accessing to the active side, maybe it doesn't really do anything. So its combination obviously works, and then eventually actually the KM drops drastically, and the KCAT is still there to be a very the enzyme. So that's. So this enzyme is a kind of like Chris brought to my lab, and I actually started to like it because I usually work on the metalloenzyme. Metalloenzyme is kind of interesting because it's very versatile. They does lots of different reactions. You can put the different metals and it does different reactions, but you can't see anything. It's very difficult to react structure to analyze it because you can't capture the intermediate of the reactions. I'm sure Sophie agrees with it. But this enzyme has intermediate in chemical reaction. Okay, so what this enzyme does is set in this classic serine hydrolase, serine attacks to the ring, and it's make the cobalt intermediate, and then water has to come in to release the rest of the product. Okay, so that means is you can capture certain step, and you can actually calculate sometimes in the step in between. So what we try to do is to try to see the bit of the reactions. I'm not going to show you everything, but this is an interesting measurement. Actually, we try to see how the tryptophan signal, so the conformational change in the enzyme, happen during the reactions. Okay, so we believe this is kind of associated with substrate binding. Enzyme is still behaving weird way. This is actually the biphasic things. We just cut the time here, but fast binding happens, and the slow conformational change is happening in second phase. So you can see the water tap has a very, very slow transition here. After the water evolution, you can see the time scale is completely different here. There's a very quick binding going on. So you can see why the KM is lower, because this is really reactive substrate. And you can see this kind of interesting plot. This is the KCAT of KM here. And this is the kind of binding constant you can see in this structure change. So there's the first two, and actually binding is slower than the KCAT of KM. So it takes time to find the substrate, and the action doesn't really matter because the accessing substrate is everything. But the last barrier in the Q4 actually binding is faster than the reactions. So the actually rate limiting step change because of those mutations. So the fast mutations obviously needs to improve the binding to recognize the reactions. That's what we're seeing. So the kinetics actually is some way showing some interesting things, and we obviously want to see some structural effect. So Chris doing the crystal structures is not really well-behaved enzyme. Some mutants, we are not able to get the crystal structure. Sometimes we get crystal structure with substrate, intermediate state, sometimes not. So there's a kind of patchy crystal structure. So then we went to the MD simulations, making the AC bond the intermediate, and then just run the MD. So it was a patchy structure data, but I can show what's happened. So 72 is kind of easy to explain. This mutation obviously includes the dynamics of the loop. This famous omega loop, and it's like determining the substrate access. So this mutation actually includes the dynamics of the loop and seems to be consistent with accessing substrate. So it kind of opens up, substrate can now get in and do catalysis. Other mutation happens when you look at the dynamics. We really don't see the much differences. So the catalytic residues, catalytic mutation happening in besides 72, we don't see the structural differences. So then we went to the MD, we done MD, we don't see it much differences. We done different analysis. This is the kind of maximum things we see some differences. So this you can do the analysis called the core variation analysis. So what I shown here is center is catalytic residues of serine 70. And when you do the MD simulation, you just vibrate all these structures. And then you can calculate how, which position is kind of moving together. Okay, so this core variation happens, you have kind of connections. If not, and then just random things happening, this distant structure is probably not doing anything together. Then this plot is actually looking at wild type over mutants, kind of noise over noise. So maybe it's nothing we're seeing, but you can see here the 72, actually a connection started to happen this side. This is one of the catalytic is kind of connecting this side, and somehow putting a lot of mutation. It's like a coordination happens during the structure. So the wild types actually behaving upside is behaving opposite way, and the lower side is behaving more like. So this kind of maximum we can see it. So the catalytic residues in effect is very difficult to see it, but the dynamics obviously important for this. All right, so that's the summary. I think I already said it, so we just skip here. So the entire summary. So hopefully you agree that this species is everywhere, and it's obviously important to understand the protein evolutions. But still it's very look like system dependent. Most likely we have to understand every system deeply, and then probably we can talk about anything because any interaction is important, intermolecular, intramolecular, long distance dynamics, and probably most important things is interaction to the substrate, and probably we had to see even the catalytic steps. So that means we had to have more examples. If you're interested, then just join us and then do more experiments. So there's acknowledgement. So there's the 3 people done is most of the work, and I presented. Then some collaborators and international collaborators, and thank you for your attention.